Good day, beautiful people. I hope you're good. I'm doing great. I am Hanifate Abelia, one of the member of Group 5, and we're going to discuss the Lesson 5, the aspects of gene therapy. So the other members are Agot Sandria Jean, Hirda Kiana, Ronyola Nico, Sayed Ryan J, Guillermo Debril, Balbuena John Marl, and Kachela Robinson. So let's start. The lesson objectives is at the end of this lesson, the student should be able to describe gene therapy and its various forms and assess the issues, potential benefits, and detriments to a global health. So first, let us know what is the meaning of gene therapy. Gene therapy involves altering the genes inside your body cells in an effort to treat or stop a disease. Genes contain your DNA or deoxyribonucleic acid. It involves altering the genes inside your body cells in an effort to treat or stop a disease. An underlying principle of gene therapy is the transfer of genetic material to specific cells of a patient in an effort to initiate a biological response to fight or eliminate a disease. So as you can see, a cell with non-functioning genes added by functioning gene, it results to a cell functioning normally. So it's either to treat, to fight, or stop a disease. Then the introduction. Medical science has detected many human diseases related to defective genes. This type of diseases are not curable by traditional methods like taking readily available medicines. Gene therapy is a potential method to either treat or cure a genetic-related human illnesses. So as you can see here, this is the human diseases, and so there are a lot of human diseases, and this type of diseases are not curable by traditional methods like taking readily available medicines. The traditional medicines refers to health practices, approaches, knowledge and beliefs incorporating plant, animal, and mineral-based medicines, spiritual therapies, manual techniques, and exercises applied singularly or the combination to treat, diagnose, and prevent illnesses or maintain well-being. A team of researchers at the Harvard Medical School in 2015 and the Boston Children's Hospital. So this is the Harvard Medical School and the Boston Children's Hospital stated that they were able to restore basic hearing in genetically deaf mice using gene therapy. The research team also reported that they have restored a higher level of hearing down to 25 decibels, which is actually equivalent to a whisper. Massachusetts is the most populous state in the England region of the United States. Eye and ear was identified as ANC80, which enable the transfer of genes in accessible otor hair cells when introduced into the cochlea of the year 2017. In 1971, human gene therapy was actually first realized when the first recombinant DNA experiment were planned. It can be simply viewed as insertion foreign DNA into a patient's tissues that hope to successfully eradicate the targeted disease. It occurred over the last 20 years was actually inspired by success of recombinant DNA technology. Without a doubt, gene therapy is the most promising yet possibly unfavorable medical field being studied.
Good day everyone, my name is Robinson M. Kachela and I am going to report the basic process in Lesson 5, the aspects of gene therapy. The basic process: replacement of mutate gene that causes disease with a healthy copy of the gene. Inactivation of a mutate gene that is functioning improperly. Introducing a new gene into the body to help fight a disease. In general, a gene cannot be directly inserted into a human gene or cell. A gene is inserted into another gene using a carrier or vector. At present, the most common type of vectors are viruses that have been genetically changed to carry normal human DNA. Viruses have evolved a way of encapsulating and transporting the genes to human cells in pathogenic manner.
Good morning everyone, my name is Kiana Dihuda and today I am going to discuss my topic which is all about gene therapy. So what is gene therapy? So gene therapy is based on correcting a disease at its root. So it is a way in fixing the abnormal genes that appear to certain diseases. So from the word itself, gene, meaning it is hereditary, and from the word itself, therapy, meaning it is something that is used to cure or to treat any certain diseases. Now I will be talking about the two types of gene therapy. The first type is the somatic gene therapy. So the somatic gene therapy is the manipulation of genes in cells that will be helpful to the patient but not inherited to the next generation. So this is according to BSCRIM 1988. The second one is the germline gene therapy. It involves the genetic modification of germ cells or cells that will pass the change on to the next generation. So the difference between these two types of gene therapy is that the somatic gene therapy is the manipulation of genes in cells which is not inherited to the next generation, while the germline gene therapy is the modification of germ cells and it will pass the change on to the next generation. So somatic, it is a manipulation and it will not pass into the next generation, while the germline is the modification and it will pass into the next generation. So to add more about these two types of gene therapy, the germline gene therapy is when DNA is transferred into the cells that produce reproductive cells. Eggs, may it be eggs or a sperm cell. So eggs are for girls and sperm are for boys. So while the somatic gene therapy is when DNA is transferred into body tissues. So what are these body tissues? For example, the muscle tissues, our nervous tissues, and more. So that's it for my discussion, and now I'm going to hand you over to the next reporter. Thank you.
My name is Janmar Balbuena, reporting stem cells therapy. Stem cells therapy are mother cells that have a potential to become any type of cells in the body. One of the main characteristics of the stem cells is their ability to self-renew or multiply while maintaining the potential to develop into other types of cells. Stem cells can become cells of the blood, heart, bones, skin, muscles, brain, among others. There are different sources of stem cells that all types stem cells have the same capacity to develop into multiple types of cells. Stem cells are derived from different sources, two of which are embryonic.
Hi everyone, my name is Gabriel Hiromo. My topic is the somatic stem cell. Somatic stem cells are the cells that exist throughout the body. Mga mga cells na exist throughout sa itong lawas. After the embryonic development, makita the different kind of tissue. Kaning stem cells has been found in tissues such as sa brain or sa utak, like bone marrow, blood vessels, skeletal muscles, skin, and the liver. They remain in non-dividing state for years until activated disease or tissue. Magpabili ni siya wala na bulag-bulag hantod nga ma-activate ni siya sa mga sakit or disease or tissue injury. These stem cells can be divided. Kaning stem cells daw pwede ma-divide or ma-self-renew indefinitely, enabling them to organ or even regenerate niya the entire or original organs generate throughout that adult or somatic stem cells are limited. Throughout the adult daw kaning somatic stem cell kay limited ra ang ilang ability to differentiate based on their tissue of origin, but there is some evidence na ebidensya na suggest that they can differentiate na to siya to become other cell types. So adult stem cell na siya blood, muscle, skin, heart siguro ni mura ni tanan. That's all. Thank you.
Good day everyone, my name is Ryan J. Desire from BSCRIM, and today I'm going to talk about bioethics. Now, before we begin talking about bioethics, we must know what bio and ethics stands for that makes up the bioethics. Now, bio in the dictionary means life, human lives, animal lives, plant lives, environmental lives. That's what bio in the dictionary means. Now, ethics. Ethics is the study of questions of morality, to know, understand what is right, wrong, and good or bad. Now, let's proceed to bioethics now that we understand what bio and ethics are. Bioethics is the study of ethical issues emerging from biological and medical advancements, questions, issues, and controversies. Like, is it right if we use a living being as a subject to test if a new medicine is working? From there, more questions will pop up, such as questions concerning its side effects, like is it harmful, or are there any side effects concerning our allergies with a new medicine? There are a lot of things to know about a newly developed medicine, but I think we should question first if practicing on living beings is morally right. That's my opinion. Now, before I proceed to the next topic, you can take notes or a screenshot on this slide. Now, let's proceed. Bioethicists. They are the professional individuals that study and conduct research concerning ethical, social, and legal issues arising in biomedicine research and advancements. Now, let's talk about the relation between bioethics and bioethicists. Bioethics establish a mutual understanding for multiple perspectives. It uses its ways to give an equal and fair consideration regarding issues that concerns human, animal, and environmental rights. It helps us know that it's not just your moral question, but others as well that is concerns with the ethical questions. And it lets your questions will be voiced out among the other ethical questions, so that the bioethicists will reach the bioethicists. Now, bioethicists are concerned with ethical questions. It appears in relationships among sciences, biotechnology, medicine, politics, law, philosophy that concerns with life. Bioethicists are the ones who study questions and look for an answer. Meaning, if there are questions or issues regarding a newly developed medicine or biological breakthrough, there would always be questions, cases for that. So those bioethicists are ones responsible for seeking answers to those questions so that it may satisfy the concerns among those questions. Now, before I proceed to the next topic, you can take a screenshot of the next 2 slides for notes. Now, let's proceed. I'm going to talk about the 4 principles of ethics. The 4 principles are as follows in the picture: autonomy, beneficence, non-malfeasance, and justice. Now, autonomy is the rights of an individual's rights to make his or her own decisions that concerns with her own health, and that right should be respected. Now, it's very self-explanatory that autonomy is a person's or individual's right, and that right must be respected regarding their health, of course, in this case. Now, beneficence states that healthcare providers must strive to improve in order for their parents to benefit. Now, beneficence is instigates those healthcare providers to improve their system so that those who are using the system will benefit even more. Now, non-malfeasance. In accordance to beneficence, healthcare providers must not harm their patients. It's pretty self-explanatory. And lastly, justice, as well as in accordance again to beneficence. Patients must share an equal amount for the benefit they receive from the healthcare system, meaning that they should put funds to their healthcare provider that they're signed up with, so that they will receive also an equal or fair amount of benefit from that healthcare provider. And that's all for my report, and the rest will be for the next reporter.
Good day everyone, I am Nico Brunella from Group 5 and I'm about to report the scope of bioethics and the 5 major issues. So what is scope of bioethics? It is artificial reproductive technologies. It has stem cell research and human genome project. It has a possibility of successful reproductive cloning, and it has synthesis of new life forms and pre-implantations of genetic diagnosis. So let's go to the five major issues. So what is 5 major issues? 5 major issue means important or serious topic or problem for discussion. So this is the 5 major issues. First one is abortion. Abortion is a removal of fetus or embryo from a woman's uterus that results in a termination of pregnancy. And the second one is euthanasia. Refers to the act of purposely ending a life to eliminate untreatable suffering. The third one is eugenics, a type of DNA manipulation that creates selective breeding to improve the human race. And the fourth one is human cloning, the scientific production of an identical human cell, tissue, or an even entire body. And the last one is the stem cell research, the area of research that studies the properties of stem cells and their potential use in medicine. So that's all for my reporting. God bless all, and thank you.
Good day everyone, I am Agot Sandrea Jin and I am here to discuss the summary of lesson 5, the aspects of gene therapy. So, gene therapy is a method that may treat or cure genetic related human illnesses. So there are 2 forms of gene therapy. One of these is the somatic gene therapy, which it involves the manipulation of genes in cells that will be helpful to the patient but not inherited to the next generation. So in short, only the patient has the opportunity to grab this kind of therapy, while on the other hand, the German gene therapy, which it involves the genetic modification of germ cells or the origin cells that will pass the change to the next generation. So in this case, both the patient and the next generation has the opportunity to grow up with this therapy since it is inherited or passed down to the sperms or manipulated to the sperms or egg cells. And there are many ethical issues on gene therapy. Some of these issues are about questions on whose authority or power to decide which human traits should be altered, and other concerns are on the discriminatory effects of those who may not or cannot avail gene therapy. So let's proceed in somatic mutations. It occurs on non-germline tissues and it cannot be inherited since only the patient is cured in this therapy, while on the germline mutations, it is present in egg or sperms and can be inherited since the eggs and sperm are mutated and transferred to the child. So in this case, since the egg and sperms are mutated, so all cells are affected in offspring, and that is why it is heritable by the next generation. And let's move on to the last question. So the question here is, identify relevant global current challenges or issues or problems and present solutions on how to address these issues. So we have here two problems, and these are targeting the wrong cells and infection caused by the virus. So when you target the wrong cells, viruses can affect more than one type of cells. So it's possible that the altered viruses may infect additional cells. So not just the targeted cells containing mutated genes. So if this happens, healthy cells may be damaged, causing other illnesses or disease such as cancer. And the other one here is infection caused by the virus. So it's possible that once introduced into the body, the viruses will recover their original ability to cause disease. So, and we have here two solutions. It is can do one several things depending on the problem that is present. They can replace a gene that causes a medical problem with one that doesn't, and also add genes to help the body fight or treat disease, or turn off genes that are causing problems. And the other solution here is the gene therapy clinical trials underway in the US are closely monitored by the FDA or the Food and Drug Administration and the National Institutes of Health to ensure that patient safety issues are a top priority during research. So that's all. And before we end our discussion, we would like to thank everyone for listening attentively to our lesson, and especially to our teacher, Miss Marisa P. Generale. Thank you, ma'am. And I think that would be all. So God bless, thank you, and have a great day ahead.